Welcome to This Explainer. Today, we're diving into one of the absolute greatest mysteries in modern medicine, the biological detective story of psychiatric disorders. For decades, the biological mechanisms behind conditions like schizophrenia and autism spectrum disorder have been this massive black box. But thanks to some truly incredible leaps in genetic technology, we're finally prying that box open. We're going to trace the clues all the way from the psychiatrist's couch down into the microscopic code of your DNA. To make sense of this complex biological puzzle, we're going to break it down into 5 quick sections: the psychiatric diagnostic dilemma, blueprints and genetic rule breakers, common versus rare variants, autism and schizophrenia genetics, and finally, editing the brain's future. All right, Section 1, the psychiatric diagnostic dilemma. Let's look at the major roadblock doctors run into every single day. Have you ever wondered why you can take a simple test for physical ailment, but mental health conditions remain so elusive? Think about it. If you have diabetes, we check your blood sugar. If you've got a broken bone, boom, we take an X-ray. But what on earth is the equivalent for a psychiatric disorder? And that brings us to our core conflict. While physical illnesses leave very clear biological footprints, which we call biomarkers, psychiatric diagnoses rely almost entirely on observation. Right now, doctors diagnose conditions based on clinical encounters and reference manuals like the DSM-5, which is essentially just a catalog of behavioral symptoms. They have to rely heavily on doctor-patient interaction. Because we lack natural biomarkers to easily test for these disorders, tracing their biological roots is incredibly difficult. Moving on to Part 2, blueprints and genetic rule breakers. To actually find those missing biomarkers, we've got to zoom way in, from the psychiatrist's couch down to the microscopic level. Look at the actual architecture of your cells here. Inside almost every cell in your body is a nucleus packed with chromosomes, which are made up of that famous DNA double helix. Your DNA is a blueprint that continuously interacts with your environment. And while we know for a fact that psychiatric disorders are highly heritable, it's just not as simple as finding a single schizophrenia gene. This is where things get tricky, thanks to a genetic rule breaker called penetrance. Basically, carrying a mutated gene doesn't guarantee you'll actually get the disease. Penetrance is just the percentage of individuals who carry a genetic variant and actually show the symptoms. So you could carry a variant for a disorder, but because of your environment or other protective genes, you never express it. Yet you could still pass that exact variant down to your kids. And to complicate our detective story even further, genes don't operate in a vacuum. They can actually mask or modify each other. This is called epistasis. It means the effect of one gene can be totally changed by a completely different gene located somewhere else in your DNA. So mapping psychiatric genetics is literally like trying to solve a billion-piece jigsaw puzzle where the pieces actually change shape depending on what's next to them. Crazy, right? Okay, Section 3, common versus rare variants. Now that we know the basic rules, let's unmask the specific genetic culprits. Broadly speaking, scientists look for 2 types of mutations. Think of a polygenic trait like death by 1,000 small cuts with common SNPs, whereas rare CNVs act way more like a single genetic sledgehammer. Let's compare them. On one side, we have SNPs, single nucleotide polymorphisms. These are tiny, single-letter typos in your DNA. They're super common, but each one has a tiny effect and low penetrance. On the flip side, we have CNVs, copy number variants. These are massive chunks of DNA getting duplicated or deleted entirely. They're rare, but man, they have a huge effect size and high penetrance, drastically altering brain development. So you can see these genetic anomalies exist on this vast spectrum. On one end, the tiny SNP typos, moving all the way up to massive chromosomal duplications on the other end. This is exactly what scientists are looking for when they sequence your DNA. But here's the thing. Finding those tiny SNPs requires a completely different tool than finding the massive CNVs. To hunt down those tiny common variants, researchers use GWAS, or Genome-Wide Association Studies. This method is essentially scanning the entire genome for suspicious statistical patterns. Scientists take a massive group of people with a disorder, like schizophrenia, and compare their genetic markers against a massive group of healthy controls, just looking for SNPs that pop up a little bit more frequently in the patients. But here's the catch. Because these individual SNPs have such a tiny effect, and because of that low penetrance we talked about earlier, scientists literally need hundreds of thousands of participants just to find a statistically significant clue. It's a mountain of data. And even when GWAS finds a hit, it usually just points to a broad neighborhood on the chromosome, leaving researchers to figure out exactly which gene in that neighborhood is actually causing the trouble. All right, Section 4. Autism and Schizophrenia Genetics. Let's apply this genetic detective work directly to real-world conditions. What have we actually found? Well, for schizophrenia, GWAS has successfully pinpointed some incredibly specific biological pathways. For decades, doctors suspected dopamine was involved. Genetic studies finally proved it by identifying variants in the DRD2 gene, which is a dopamine receptor. We also found massive evidence pointing to glutamate and calcium signaling through genes like GRIN2A and CACNA1I. This biologically proves that neurotransmitter communication is fundamentally out of balance in these patients. Similarly, in autism spectrum disorder, we see a heavy genetic overlap with synaptic function. We're talking variants in genes like CADPS, which helps neurons release neurotransmitters, and KMT2E for chromatin regulation. But we also see devastating rare variants, like deletions in a gene called SHANK3. SHANK3 basically codes for the physical scaffolding on the receiving end of a neural connection. Without it, the physical structure of the synapse literally breaks down. Because these are incredibly complex diseases influenced by potentially thousands of genes, scientists now compile all these tiny clues into a single polygenic risk score, or PRS. By mathematically adding up all those thousands of tiny variants you carry, they can calculate your overall likelihood of developing a condition. It can even help predict treatment resistance, which is an absolutely massive step toward personalized medicine. Which brings us to our final section, editing the brain's future. We've found the genetic clues. But how do we actually study these typos in the lab? And more importantly, how do we fix them? How exactly do scientists translate these genetic clues into futuristic cures? This is where we step into the high-tech, borderline sci-fi laboratory tools of modern genetics. Finding a gene is just the beginning. You have to see it in action. This process is mind-blowing. First, you replicate mutations in mouse models. But then it gets wild. Using iPSCs, scientists can literally take a basic skin biopsy from a patient, reprogram it back into a stem cell, and coax it to grow into a living human brain cell right there in a dish. Then, using the molecular scissors of CRISPR-Cas9, they can edit the DNA to fix the mutation and watch exactly how the brain cell changes. And then there's optogenetics, arguably one of the greatest breakthroughs in modern neuroscience. We are literally borrowing light-sensitive proteins from algae and inserting them into mammalian neurons. Once they're in, we can control the brain with light. Shining a blue light opens ion channels and forces the neuron to fire. A yellow light forces it to shut down. We can turn specific brain circuits on and off using tiny LED lights. So I want to leave you with this provocative thought. If we can control neurons with flashes of colored light and edit DNA at will, how long until psychiatric disorders are a thing of the past? We've gone from diagnosing patients just by talking to them to growing their neurons in a dish, editing their code with CRISPR, and controlling their circuits with light. With these futuristic tools rapidly advancing, are we finally on the verge of solving the ultimate biological mystery? Thank you so much for joining me for this explainer. Keep learning, keep questioning, and I'll see you next time.